Stephen Stamp, CEO and CFO of Midatech Pharma PLC. If I may, I'll leave the disclaimer as read and get straight into it. Midatech is a biotech company. We're focused on drug delivery. I'm going to start up with a confessional slide here because the company has had a little bit of a checkered history and, in my opinion, has made some missteps in the past, which I'm here to tell you that my job is to correct for the future. So the company IPO'd back in 2014 on the back of some gold nanoparticle technology. The initial experiments weren't apparently terribly successful. And so the company tried to remodel itself as a specialty pharma company and bought a business in the US called Dara Biosciences in 2015. The idea was that that was going to contribute cash flow and earnings and help fund the pipeline, and eventually would be the marketing outlet when the products matured and were ready for promoting in the United States. Unfortunately, that wasn't a terribly well constructed acquisition, and the company made losses from the outset, and it got worse from there. And so it was sold at a significant loss in 2018. The company then looked for its next nearest program in terms of proximity to market, and that was a program called MTD-201, which was designed to be a knockoff of a Novartis product called Sandostatin that had been on the market for a number of years, about $1.6 billion in sales, was off patent, but nobody had ever been able to launch a generic version of it. Well, this company had in its portfolio something called the Kustera Technology and thought that it could mimic the Novartis product relatively easily. In fact, what they developed was a much better product than the Novartis product, and I can show you that in a minute, but it was not bioequivalent to the Novartis product and therefore would have required a full clinical program at a cost of about $30 million, including the building of a bespoke factory in Bilbao, Spain. Well, COVID hit, we struggled to raise money. The licensee that we thought we had for that program pulled out. And so, frankly, we had no choice but to reorganize the company. We shut down the Bilbao operations, halved the cash burn rate from over $1 million a month down to a bit less than $500,000 a month now, and went for something called, and I borrowed this phrase when I was in America, a multiple shots on goal strategy. And by that I mean, instead of focusing on one product and trying to get it to market ourselves, almost an impossible task for a company of our size and resources, what we decided to do was work on multiple products at the same time, but at a much earlier stage, with a view to partnering them off at the appropriate time to large pharma, who would then take the product on through clinical trials, and we would get royalties and milestones. Obviously, we're giving up quite a lot of the upside, but we're also increasing our chances of partnering off some of these programs. So the company has 3 drug delivery technologies: Kucera, Midasolv, and Vitacore. There's a common theme in that they are all designed to improve the biodelivery and biodistribution of medicines, already approved medicines. We're not in the business of inventing new medicines. We're in the business of improving existing medicines. So what that means is our technologies help deliver the medicines to the place in the body or the site of action where they're most needed, instead of having them washing around the system, delivering side effects and unwanted toxicities. And this is particularly important, for example, in oncology products. So here's our pipeline, a total of 9 programs, and the progress we've made in the first half of this year is we've advanced to our internal Kucera programs, 211 and 219, into preclinical phase, and we have preclinical data I can share with you in a minute. We've also delivered 2 proof of concept molecules to our partner, 214 and 216, for them to take into preclinical studies themselves. And our Midasolv program, which is focused around a molecule called panobinostat, we've also advanced that, and we're about to start a phase 1 study in GBM, glioblastoma, which I'll talk about in a second. And then we've also dipped our toe back into the original technology of this company, the gold nanoparticle technology, with a psoriasis product. Again, more of that in a second. So let's start with our clinical programs and MTX110. So we've refocused MTX110 to focus on GBM or glioblastoma multiforme. This is an intractable form of adult brain cancer. It always leads to death in the range of 13 to 30 months, depending on the genetic profile of the patient concerned. Absolute tragedy. There are no really useful treatments for this condition. There are one or two old molecules that are analogs of, believe it or not, World War I mustard gases, which bring with them their own side effects. A lot of the drugs that have been developed for this area don't work because they don't cross the blood-brain barrier. The blood-brain barrier is very effective keeping toxicities out of the blood, out of the brain, not surprisingly. We are using a molecule called panabinostat, and we've got some quite good preclinical data as illustrated by these charts on the right here to show that it is an effective killer of patient-derived cancer cell lines in GBM. The trouble with it is it's insoluble, so our technology has made it soluble. And secondly, it doesn't readily cross the blood-brain barrier. So what we're doing is we're deploying a pump and a catheter, and the catheter is inside the body, goes up through the neck into the skull. We can deliver the drug directly to the tumor, and by doing that, we think we can get about 100,000 times the concentration of drug in the tumor compared with what you would get if you took the product orally. And of course, because the drug is being directed to the tumor rather than systemically, you're avoiding a lot of that toxicity you would get with systemic application. So we have an IND filed. We're currently answering FDA questions. We had a very positive meeting with them, and we expect to start the trial right in the new year. So we're quite excited about that. And if we get a signal, those data will be available sometime in the early fourth quarter of next year. There is a wrinkle here, though. Panabinostat is still under patent, and the license holder has actually terminated our license. We're not too sad about that because the royalties and milestones in that license were frankly not commercial, were negotiated by this company some time ago, and the patents will run out before this product is ready for market anyway. However, it may cause a slight delay in the second indication called diffuse intrinsic pons glioma, or DIPG. This is a childhood form of brain cancer, and it is also intractable and also always leads to death, usually in this sort of 10-month range again. The difference here is that these tumors are non-resectable because they're sort of diffused in the brain stem. The picture on the right there, which is it's an enhanced image of the tumor. It shows you how we're managing to get the drug using that same catheter pump system into the tumor. We have phase 1 data on this program, which were announced last October. Excuse me, 2 Octobers ago now, October 20, and we showed that we were able to increase survival only in 7 patients, I must add, but we increased survival to 26 months compared with 10 months in the background information. So we think this has real potential here too. This is an ultra-orphan indication, whereas GBM is a $5 to $7 billion opportunity. This is probably a $100 million opportunity, relatively few patients, and difficult to recruit for. So let's move on to Kucera. This is the sort of main technology in the company, based in our facilities down in Cardiff, where I am today. It is a method of 3D printing drugs. So what we do here is we combine a drug with a polymer and a solvent, and we use a printer. It's an illustration of how the printer works. You can see the droplets there on the top left picture, with an industrial print head, and we fire those droplets into a stream of anti-solvent, and the droplets are then immediately formed into microspheres, something like 30 to 50 nanometers across. And there's a picture of one on the bottom left there, split open. And those nanospheres contain drug, and they're remarkably monodispersed. So they're of the same size, a small size, and they contain a uniform amount of drug. What that means is that they can also be tuned. So we can deliver these microspheres via injection into a depot in the body. They then biodegrade over time, releasing drug from a few weeks, maybe up to 6 months. And we can get very nice, predictable, flat pharmacokinetic profiles, which you wouldn't get if you took a drug orally or via systemic injection, because you get troughs and spikes and troughs. Spikes when you give the injection and a trough in between injections. We get much more sustained drug delivery. So these three charts here illustrate how we can change the amount of drug per microsphere. We call it drug loading. We can get up to about 20% in total. We can attenuate the burst release. So that's the immediate burst you get with a drug delivery by lowering that or increasing it as required. And because of the uniformity of our microspheres, we get good injectability. So with some of the competitor products, for example, about a third of the time their needle clogs and the process has to be repeated. And those injections can only be given intramuscularly, usually in the backside. Because ours microspheres are smaller and the viscosity is much lower, we can use smaller gauge needles and we can deliver the drugs subcutaneously, which means it could ultimately be delivered by the patient in the home rather than in the doctor's office, again, taking cost out of the system. So there are many features and advantages to the Kustera technology which are listed here. What I wanted to draw your attention to in particular was a new area of exploration. Oops, I lost the presentation again, sorry. And that is we are using the Kustera technology to target tumors. So we can now deliver the drug directly into the tumor rather than subcutaneously or intramuscularly. So all the drug is in the tumor, and we can hold the microspheres then degrade over a period of time, effectively holding the drug in the tumor while it does its work. If you don't use a system like ours and just inject drug directly into the tumor, it is rather rapidly eliminated, partly because of the vasculature of the tumor and partly because of the fissures and crevices and the like, which means it gets eliminated rather quickly and doesn't have a chance to have the impact you might hope it would. So here's the clinical validation of our system. I mentioned MTD201. This is the product. On the left-hand side, these are individual patients, and you can see how uniform the individual patients are in terms of pharmacokinetic blood levels over a period of time. On the right-hand side, this is what the same active looks like, but in the Novartis system, product called Sandostatin, and you get a lot of interpatient variability. Now you can understand why we were unable to copy directly the Novartis product. We have a much better product, much more predictable, and much less interpatient variability. The downside of that was it required a full development program. So here's some data from under our new strategy, multiple shots on goal. The first program we're working on with our Kustera technology is on a molecule called Brexpeprazole. If you don't know it, I suspect you won't, it is the latest generation of antipsychotics products. All of this market, with the exception of Brexpeprazole, has moved to long-acting products. And that's mainly for patient compliance reasons. If you have a psychotic patient, you don't want to have to rely on them to take tablets every day. You want to call them into the doctor's office and give them an injection which lasts them for 3 or more months. Here are some data on the slide. The blue line represents the blood levels of Brexpeprazole using our Kustera technology, and you can see they're nicely sustained over a 90-day or 3-month period at therapeutic levels. This compares with the immediate release Brexpeprazole, which is the orange line, which quickly goes below therapeutic levels, which is kind of what you'd expect. So here we are. We think we've got data which shows we can deliver a 3-month product. This product is available for licensing now, and we have a number of conversations ongoing. The second product that we're working on in our in-house portfolio is called Tacrolimus. It is indicated for prophylaxis of transplant rejection, a bit like cyclosporine. the difficulty with this particular product is it has a relatively narrow therapeutic index. So if you give too much of it, you get liver toxicity and oversuppression of the immune system. You give too little of it, and you get transplant rejection, both of which are bad outcomes for patients. So it's a little bit more difficult to tell with this chart, but it looks like we've probably got a 3-week product here. This product is currently taken twice a day. So this will be a major convenience for a patient, but also it probably needs one more round of formulation work here. But if we can deliver a product which keeps it within that narrow therapeutic index for a period of 2 or 3 weeks, we think that might have major clinical advantages for the patient too. So those are our 2 internal programs. The big news that we announced in the middle of this year, we think this is a world's first, is that we have been able to encapsulate a protein using our Microsphere technology. As far as we can tell, nobody else has ever been able to do this for a couple of reasons. One is the proteins are incredibly fragile, so easily denatured, and once that happens, they're no longer able to perform the function they were designed to do. And secondly, most of the competing technologies either involve heat or shear forces or toxic solvents. All of those can effectively denature the protein too. Our manufacturing process, as described for the printing, is relatively benign, and we were successful this year in actually encapsulating the monoclonal antibody, which is a very large protein, about 150,000 kilodaltons, 150 kilodaltons, without denaturing it. So what? Well, no monoclonal antibodies currently are available in long-acting formulations. They all are in immediate release, which means you get the peaks and troughs that I was talking about earlier. The total sales for the top 10 monoclonal antibodies are about $75 billion, and over 150 for all of them put together. So there ought to be opportunities in there to apply our technology, both to improve the pharmacokinetics of those products and offer life cycle extension opportunities using our technology. So how do we prove it? Well, these 3 charts here kind of demonstrate it. On the left-hand side, we heated a monoclonal antibody and found that we got 0 inhibition relative to the antigen, which is kind of what you expect because we destroyed the effectiveness of the antibody. We then took the antibody untreated and got 100% inhibition, which is the middle bar, positive control on the left. So we now know that the antigen test works. And then we formulated the antibody in Kustera technology, and we also got 100%. So now we know that we've actually been able to retain the inherent activity of the protein inside our technology. The middle chart is kind of the same thing, but instead of having 0 or 100%, we increased the concentrations of the protein in Kucera from left to right, and we got increasing amounts of inhibition, which is kind of what you expect, a linear response. And on the right-hand side, I'm illustrating here the release profile over 6 weeks of the protein leaching out of the microspheres.
Stephen, it's Chris here.
Yes.
I was going to say we've got a few questions coming through, so you'll need to wrap up pretty soon if that's okay with you.
Last slide. Lastly, the Vitacore technology, we've investigated that, as I was explaining, with methotrexate. The 3 pictures on the right-hand side illustrate the inflammation in the mouse model using a control or Vaseline and Divabet, which slightly decreased inflammation, and our product, methotrexate, combined with gold nanoparticles, less still. So we're quite hopeful of this program, and we're in the process of repeating these experiments. Okay. I'll just tell you about things we achieved in the first half, which I've already talked about. So I'm ready to take questions, Chris.
Firstly, would Novartis be interested in MTD-201, given it was working with and improving on what was originally their drug?
Not surprisingly, we showed it to them, and they have effectively given up on the drug. They've assumed it's going to be generic, and have no interest in trying to rescue it.
Okay. With Q-Sphera, do you investigate additional polymers to see if they offer any further benefit, or do your existing polymers work just fine?
So we have a range of polymers that we choose from, and we have a range of solvents that we choose from, and there are different pressures and temperatures and anti-solvents. And those are basically the ingredients that we use to tune the system to either get it to release slower, faster, attenuate the burst release, etc.
Okay.
But that's all, that's kind of know-how. We don't publish what they are, as you might expect.
Yeah. What else can be said about the Kucera bio litigation? Question mark. Will it be resolved in court with consequent costs and risks?
So I haven't got much time here. They basically terminated the license on the grounds that we were allegedly proposing to liquidate the company, which was frankly ridiculous. By the time they posted that notice, we'd already raised money. But they chose to construe, some odd phrases in various press releases and terminated the license. As I say, we weren't too unhappy about that. And then a year later, literally a year later, they terminated the license again, this time on grounds that for material breach, because we hadn't kept them informed of what we were doing. Well, of course we hadn't kept them informed because they terminated us a year earlier. The reason they tried to terminate us a second time is that for material breach, they would be entitled to a license to our IP. And there is no way on earth we're going to be giving them a license to our IP. So we're trying to sort of keep the cost to a minimum. We push back on all these, frankly, what we regard silly letters they send us. And they've been quiet since August now. But if we have to go to court, we have to go to court.
Okay, thank you. If you were successful in partnering MTX 110, would it likely cover both GBM and DIPG?
Yes, because the dosing and the delivery system are very similar. So it would make sense to package the two together. Otherwise, you'd sort of be competing. It'd be hard to track sales and for milestones and royalty purposes.
Fair enough. What would you regard as a successful 2022, i.e. how many partnerships should we expect to see from you?
Yeah, so I'd like to see one partnership in our large molecule protein work. I'd like to see one small molecule partnership, which would either be Brexpeprazole or Tacrolimus. And I'd like to see positive phase 1 data from our MTX 110 program in GBM. If all of those three came off, that would be quite transformational for our little company.
Excellent. And time for one more quickly, Stephen. Data readout is 2 months old per timeline given. What is the delay?
Just recruitment. Both of these are orphan indications. It takes a while to find the patients, and then you have entry criteria. It's just a process. COVID hasn't helped, of course, because there are limited numbers of surgical centers that can handle these types of complicated surgeries. You can imagine you're implanting a catheter in somebody's brain. You've got to fly patients around. And then, when lockdown and stuff, it's not easy.
Stephen, that's fantastic. Thank you very much indeed this evening.